Good morning and welcome to the IHI Call Days Call 5. My name is Sergio and I will be moderating this info session. Today's session, it's called Accelerating the Implementation of New Approach Methodologies and Other Innovative Non-Animal Approaches for the Development, Testing and Production of Health Technologies. Before we start, I would like to read an important notice. All information regarding future IHI call topics is indicative and subject to change. Final information about future IHI calls will be communicated after approval by the IHI Governing Board. We are recording this session and it will be published on the IHI website and the B2Match platform. We will also publish the presentation slides. If you have questions, please use the chat box that you will find on the bottom right of the screen. Without further ado, I would like to introduce you my colleague Salome, who is a scientific officer at IHI and will be presenting today's topic. Salome, the floor is yours.
Thank you, Sergio, and welcome everyone in today's session. So today we will cover we will do an introduction to the IHI program, and then we will talk about the topic text, and more precisely, we will talk about the challenge, the need for public-private collaborative research, the scope, the outcomes and impacts, as well as the budget. We will provide information on proposal submission and evaluation, and we will provide some tips for writing a successful proposal. This session will not cover rules and procedures and how to prepare the financial proposal. So these webinars are on the IHI website. And if I'm correct, the rules and procedures took place yesterday, and the preparation of the financial part webinar will take place at the end of this week. So a few words about the Innovative Health Initiative. We are a public-private partnership between the European Union, represented by the European Commission, and the healthcare industry associations, represented by COGSIR, which is focusing on medical imaging, radiotherapy, health ICT, and electromedical industries. EFIA, including Vaccines Europe, focusing on pharmaceuticals and vaccines. Europa Bio, on biotechnology, and medtech technology, medical technology industry. So IHI has several objectives, but the most important one is to try to turn health research and innovation into real benefits for patients and society. And to do so, we need to foster the delivery of safe, effective health innovations that cover the entire spectrum of care, and especially in areas where there is an unmet public health need. Of course, we want to make Europe's health industry globally competitive. And on the bottom of this page, you can find a link to the SRIA, so the Strategic Research and Innovation Agenda, which is basically a document that details the areas IHI should work on and helps to guide IHI's decision on which ideas should be then turned into call topics. So, as you might already know, this the IHI funding model is it's a very specific one. So we are a public-private partnership, which is funded by the European Commission through a cash contribution, and this is primarily used to support universities, research organizations, patient organizations, SMEs, and mid-sized companies. In single-stage calls, there is also possibility for large companies to receive EU funding. But I recommend you watch the rules and procedures, the webinar on rules regarding this call, so you can get more information. So as we said, part comes from the EU cash contribution, and the other part comes from the IHI Industry Association and contributing partners. So you must provide at least 45% of total project eligible costs, and this is comes usually in the form of researchers participating in the project, infrastructure, and so on. So this is a single stage call, which means that when the call is launched, it is open to consortia that have to include from the start all the actors that need to be together to implement the activities and to lead to the success to the specific project produced by the proposal. So these consortia, which include usually private partners, academics, contributing partners, SMEs, healthcare professionals, regulators, patient organizations, and so on, need to prepare and submit a proposal. All the submitted eligible proposals will undergo a competitive evaluation by the independent experts, and there will be a ranking list. And as long as there is budget and the proposal is above threshold, there will be the possibility to get funded. Once the proposals are evaluated, there will be a ranking for the proposals above threshold, and based upon the availability of funds, there will be one or more proposals that will be invited for the preparation phase of the grant agreement. And if everything goes as wished, then they will be invited to sign the grant agreement and start the project. So now I'm gonna go through the details which are included in the topic text. And as you already know, this is a single stage topic, which means that it is broad in scope. Nevertheless, there are precise points on expectations about what applicants should consider when preparing the proposal. So please read the topic text very carefully, as this will be the basis for the evaluator's assessment. So what is the challenge that this topic wants to address? So animals and animal-derived materials are widely used in biomedical research and in development and production of health technologies. And this raises ethical concerns, and there is also a growing societal pressure to move towards alternative approaches. And there is also a legal obligation to replace, reduce, and refine the use of animals in research, the so-called 3 Rs. So besides ethical concerns, there is also scientific evidence that supports moving away from animal-based approaches and finding more human-relevant methods and strategies for both the assessment of safety and efficacy of new health technologies. Animal testing requires time-consuming protocols, high cost for animal supplies, and the results are not always reproducible and applicable to humans. In addition, for the development and production of health technologies, such as in vitro diagnostics, as well as in biomedical research in general, materials of animal origin are required, such as biomolecules and sera. So these animal-derived products requires large amount of animals for their production. So the new approach methodologies and other innovative non-animal approaches have high potential to improve the development and/or the production of health technologies while contributing to the reduction and replacement of the use of animals. Although the potential of new approach methodologies and other non-animal approaches for the production, development, and testing of new health technologies, more evidence and high-quality data are required to evaluate their performance and to support regulatory decision-making. Next slide, please. Yeah, the current topic wants to address these challenges by exploiting the latest relevant scientific advancements to develop the new approach methodologies and other non-animal approaches, which could be more readily available and more efficient than the ones involving animals, and which should improve either the development, including efficacy and safety assessments, of new health technologies, in infectious or non-communicable diseases or the production processes of such technologies. Regarding the new approach methodologies, for the definitions, you can please refer to the footnote that we have in the topic text. So why this is a topic to be tackled in IHI? So the right platform is needed to achieve the expected outcomes and impacts of this topic. So to this end, an appropriate multi-stakeholder and cross-sectorial private-public collaboration is required. Partners from different types of industries are required to become partners, such as from industries like pharma, medical devices, in vitro diagnostics, vaccines. Also from academia, SMEs are all needed to collaborate to reach the objectives, which is to accelerate the development and use of effective new approach methodologies and other non-animal approaches in the testing, development, and production of health technologies. The involvement of patients, regulators, and policymakers is also needed to guide and advise on regulatory acceptance criteria, to foster acceptance and to facilitate the update or translation into health policies. So this exchange of data, expertise, and knowledge will all hopefully allow the generation of evidence on applicability of these approaches in industrial context and to support regulatory decision making. Here we would like to put emphasis on the public-private partnership IHI model. So applicants should take full advantage of the IHI model that is based on a leveraging of contributions coming from the industries under the umbrella of the association members of IHI, with the GU funding coming from the EC. So what is in the scope of the topic? Well, applicants have the liberty to choose between developing new approach methodologies or other non-animal approaches, or combination of those, or use them in an innovative way for the development, assessment, or production of health technologies, such as biopharmaceuticals, vaccines, medical devices, including in vitro diagnostics. Please specify the context of use. For example, is it for primary pharmacology, toxicology, safety, quality control, production processes of the novel approach or approaches, how it or they can be integrated efficiently in the relevant workflows, and propose and implement a plan to carry out their performance evaluation and validation, as well as demonstrate the added value in comparison with the relevant established animal-based approaches. Please generate evidence on the robustness, reliability, and applicability of these novel approaches in an industrial R&D context, and to support regulatory decision-making in testing, development, or production of health technologies as relevant. Accordingly, applicants should develop a strategy for generating appropriate evidence to support regulatory acceptance, as well as engage with regulators in a timely manner. Please gather and produce high-quality datasets to generate a solid knowledge base for supporting the use of these approaches in the field of health technology and drive 3Rs implementation. To ensure sustainability of the results and foster future development and validation of these approaches, applicants should develop a fit-for-purpose scalable digital data repository. So you will need to establish a collaboration platform between all private, public, relevant stakeholders, including regulatory agencies and policymakers. Patients or patient organizations may be included and actively contribute to such activities by providing, for example, their insight on the use of human-derived samples as relevant. You will need to plan a strong communication and dissemination plan to accelerate the implementation of these approaches, and also explore synergies and complementarities with relevant initiatives at national, European, and international level. So there are some expected outcomes from this topic, and applicants are expected to address all the points that are listed in the topic text. So researchers are expected to benefit from the implementation of new approach methodologies and other non-animal approaches, which have been assessed and validated for their performance and found to be relevant, reproducible, predictive, and standardized, ultimately leading as relevant to their regulatory acceptance for the use in infectious and/or non-communicable diseases applications. So these new approaches should lead to an improvement of animal to human translation and/or production processes and to a significant reduction in the number of animals used. The European industry will benefit from the establishment and availability of these approaches for the testing, development, and/or production of health technologies that are fit for purpose to support regulatory decision-making. Researchers and developers of innovative healthcare solutions will have access to high-quality data, new recommendations, and best practices to incentivize the use of new approach methodologies and other non-animal approaches and their integration in industrial processes. This should be supported by an appropriate digital repository to ensure sustainability and scalability of the knowledge base. So regulators and policymakers will gain knowledge thanks to this research and have access to high quality data on the characteristics and use of these approaches to foster the development of harmonized guidance and requirements, as well as uptake or translation into health policies. Projects under this topic are expected to contribute to relevant EU health policy initiatives, such as the new Industrial Strategy for Europe, the European Health Emergency Response Authority, HERA, and the European Health Data Space. So in the long term, it is expected that this research will help breaking down silos. So bring together different stakeholders to foster the use of these approaches in the efficient development, testing, and production of safe and effective innovative health technologies. And it is also expected to improve public health, so patients will benefit faster from the safe and effective health technologies developed using NAMs and other non-animal approaches that provide more human-relevant data and may be more predictive than current approaches. Also, it will foster the development of health policies and standards on the use of these approaches in health technologies. Hopefully, it will enhance the competitiveness of the European health industry as high-quality, innovative approaches and methodologies will be developed, which can reduce the time and costs of processes while reducing the use of animals or animal-sourced materials. And this will also help EU become more sustainable and autonomous by achieving regulatory validation and uptake development of such approaches that are not dependent on shortages with animal supplies. So a few words about dissemination, exploitation, and communication. So these aspects are very important throughout the lifespan of the project. And please make sure you reserve budget and resources for these activities. Then it is also very important to demonstrate, and this will be very carefully analyzed by the evaluators, that you have a good plan for dissemination and exploitation to maximize the impact. If in the topic, the 3 A's, so availability, accessibility, and affordability provisions clause is applicable, then the specific elements relevant to the clause will have to be addressed. Please note that this info will be made available in the final version
of the topic text once it is approved by our governing board. And it will be made available in our website in the submission environment. So it is important to plan communication measures for promoting the project and its findings. You will also need to provide in your proposal a short description of the dissemination, exploitation, and communication activities. And be careful, this is an admissibility condition. If you don't have it, the proposal is not going to the evaluation phase. And also consider how your plan will evolve in the project, and also submit and consider updates throughout the course of the project and also beyond it as it will continue much further. But this is something to be dealt once the project has started. A few words about the budget. So this topic has a total budget of 30 million of public funding. So while IHI estimates that 12 to 15 million would allow a project to address the outcome adequately, it is up to the applicant to arrive with its best proposal for a budget for their proposal. But this, of course, needs to be justified. It is very important that you keep in mind that it is recommended to have contributions from project participants of at least 50% of the project budget. So the eligibility criterion is 45% contribution of the action's eligible costs to be covered by the contributions from project participants. However, we strongly recommend you having 50% of the project's budget, as you might have some issues at later stages. So it's better to foresee a little bit more than 45%. So as an example, if you have a project requesting 20 or 15 euros of million euros of IHI financial contribution, the contributions from project participants will be 20 or 15 million euros, depending on what you are asking as IHI financial contribution. And these two parts collectively will be the project budget. So by project participants, we mean industry members of the IHI private members, such as COSIR, MedTech, FPA, Vaccines Europe, and Europa Bio, as well as contributing partners. Please note that for contributing partners, there are some very specific requirements which are explained in the Rules and Procedures webinar that took place yesterday, which are already available on our website, because there are some particularities to take into consideration in case you are thinking of becoming and contributing partners. So you are very, very encouraged to watch these Rules and Procedures webinar. So a few words about the duration. This is really up to you to decide, but of course will be evaluated by the experts, and it needs to be justified. So therefore you choose your duration in alignment with your project activities and expected outcomes and impacts. So please, when you prepare your work plan, try to propose something that makes sense, because this will be very carefully analyzed. So this is a simplified example budget. As we mentioned, what you have to keep in mind is that at least 45% of the total cost must be covered by contributions from project participants. And this is an example of budget because we know that it can become quite complex. So you need to understand and remember the different type of participants. We have the public partners, which can be universities, hospitals, SMEs, patients organizations, regulators, and so on. And these members, so they they request for funding their activity via the IHI funding. And then there are also members that provide contribution but do not request funding, like the industry associations and contributions. There is also possibility to have a hybrid model where the industry associations and contributing partners can both request funding and provide in-kind contribution, the so-called hybrid model. But to have an eligible proposal, you need to have contribution of at least 45%. We strongly recommend having 50% to be on the safe side. Of course, if you would like to have more information, more detailed information on the budget and on the different type of participants and different possibilities, again, we strongly recommend you watch the Rules and Procedures webinar and also the financial webinar. So a few more slides. Keep with me. I know it's a lot of information to process, but yeah, I think it's very important that we provide all the useful information that you can use to prepare a successful proposal. So a few words on the proposal submission and evaluation. So the participants can update the information in the submission system at any time before the final submission. The proposal template has part A, part B, and some annexes. The part A is where you can fill it online with, and you need to provide administrative data, who you are, etc. And the part B is the so-called narrative part with different sections, which on excellence, impact, quality, and efficiency of the implementation. All these are important because they correspond to the three equally important evaluation criteria. And there are very specific instructions to follow that you need to keep in mind when you prepare this part, and that you can find in the submission environment. Then you have the annexes with different series of annexes. The participant type, where you will need to explain the type of your affiliation and stakeholder type. Another annex where you will need to provide the budget details. Also another, the coordinator declaration. If relevant, the in-kind contribution to additional activities. And for this, please watch the webinars that we mentioned before, as we will not get into details in this one. And also there is an annex on clinical studies template. However, you need to keep in mind that if no clinical studies are included in the proposal, you will need to upload a document, which is basically a page with a statement to confirm that no clinical studies is foreseen. So now we're gonna briefly talk about the evaluation criteria. So when your proposal is found to be eligible, it reaches the evaluation phase, where it will be assessed by a panel of independent experts that have the adequate and relevant multidisciplinary expertise to judge your proposal. So we have three criteria, as we said: excellence, impact, and quality and efficiency of implementation. There is a threshold of 3 out of 5 for each criteria, and an overall of 10 out of 15. So very briefly, in excellence, you will need to justify, to explain the pertinence of your clarity and pertinence of the project's objectives. And you will need to show how ambitious it is, but still feasible, that goes beyond the state of the art, that is sound, and many different aspects that you can have a look in detail whenever you want. You also have the second criteria on impact, where you will need to demonstrate the credibility. So it's very important that you are coherent throughout your proposal, and also the suitability and quality. So for this, it's very important that you also write a dissemination and exploitation communication part appropriately. And then the quality and efficiency of the implementation, where you will have a global evaluation of your proposal of how the private-public partnership is able to deliver what is promised, the capacity, and so on. So I'm not going in detail through the sub-criteria, but yeah, please have a very good look in while you're preparing your topic text and follow, sorry, the proposal, and follow the instructions. A few tips for applicants. So as I already told you, your basis for writing your proposal is the topic text. So we are you are asked to read all the call relevant material in the link mentioned in this slide, especially the topic text. And of course, to watch the rules and procedures and the preparing the financial part of the proposal webinars. This is very important. A few additional tips. Please form your consortium early, because it takes time to write a good proposal. And to form your consortium, please start working as early as possible. Always think in a private-public way, where all the members have their role. There is an engagement in the activities and in the governance in the private-public spirit. Remember, of course, to bring the in-kind contribution, otherwise it's not eligible and you will not reach the evaluation phase. Ensure that all information requested in the call text and proposal template is provided to allow the evaluation experts to do their job. And of course, consider and plan for the potential regulatory impact of results. So I'm slowly reaching the end. Finding project partners, this is very important. There is no one magic way. You need to combine the different tools and possibilities that you have. So please network with your contacts and IHI Call Days participant. Use the EU funding and tenders portal partner research search tool. Get in touch with your IHI national contact point, network on social media. And something that, pitching, something that is very useful for you is that you participate in the pitching session. So the pitching session will take place in a few, in about half an hour. So it's today, 27th of June, from 10 past 11 to 10 past 12 Brussels time. And here you have the link where you can join. And Sergio, as I can see here, these are the names of the people participating in the pitching session. So this is a great opportunity for you to network and find potential partners for your proposal. Finally, we have put in place for you a platform, the B2Match platform, that you can use to book meetings. So here you can see that it's very easy. For easy steps, you can do this. So just make yourself available, look for partners on the participation or organization tab, because there is a possibility to select the topic, the specific topic, and see the relevant parties that they have manifested their interest. Select date, time, and attendees. You can add a message and send a meeting request. So we prepared, the communication IHI team has kindly prepared a step-by-step guide on how to book meetings. And this was my last slide. Now this is an opportunity for you to ask for questions. So if you want to ask a question, please use the chat function on the right corner of your screen. I will be happy to take your questions. I also have here my colleague Elisabetta Vaudano, Principal Scientific Manager, and we have also a lawyer, a legal officer of IHI that may also be very useful in case you have more legal natured queries. So that's it for me. Thank you very much. Sergio, the floor is yours.
Thank you very much, Salome. And now I see that we have already received a lot of questions on our chat. So I would like to encourage you to use the chat for asking your questions. And before we start, also, I would like to inform you that if you go to the platform, to the B2Match platform, and you select Marketplace, you can find for this very specific topic 31 opportunities that actually they vary from project cooperation to very different proposals. Some of them are very interesting, like the remote vital function monitoring, evaluation and smart intervention, stakeholder engagement strategies, organs on chips for advanced in vitro modeling, virtual control groups to replace current. Cannot see the rest, but you get the idea. Just go to the marketplace and look for marketplace opportunities. So let's go directly now to the questions. And the first one is from Irene. How can we identify private partners and contributing partners? Are they defined by IHI, and should they be included from the beginning in the consortium?
Okay, perhaps Desmond would like to reply to this first question.
Yeah.
Hi, everybody. Sorry, I had a. Can you guys hear me okay?
Yes, we can hear you.
Sorry, I had a problem unmuting there in the tool. So in terms of the contributing partners, a couple of points here that Salome touched on and we'll just reiterate. So the contributing partners join the actual applicant consortium, and it's up to the applicant consortium to identify these contributing partners. So by their very nature, contributing partners are not in any way connected with either the IHI joint undertaking or with the private members of the IHI joint undertaking. So it is in fact a rule that to become a contributing partner, you cannot be, one of the companies that makes up with the trade associations that are the private members of IHI. So you can't be a pharmaceutical company, and you actually can't be affiliated with them either. So, I mean, contributing partners, they vary quite a lot in terms of character, who they are, but I think that the normal example of a contributing partner would basically be something like a charitable foundation that would be coming from the US or within Europe itself, which doesn't actually have any formal association with the private members of IHI. And then, in terms of then getting these contributing partners on board, as I said, the applicant consortium will reach out to them, ask them if they want to become involved in the actual project. And then if that entity decides that they want to become a contributing partner, then they basically start the path to becoming a contributing partner by submitting an application letter to the IHI joint undertaking. So if you're contemplating using contributing partners, I would suggest the very, very first stop should be the section on the IHI website. We've got a dedicated area there which explains what exactly is required to become a contributing partner. There's a guide there as well which lays out everything from the application process through to the financial rules that apply. There's a template letter that we've updated 3 or 4 times recently, and it's actually quite a good template letter now, and it will have all the information. So I think the short answer I can give to the question is, A, the applicant consortia are finding the contributing partners themselves. We're not pointing anyone in their direction. And I think this echoes with Salome's point, that basically the applicant consortia form themselves. IHI don't put the projects together. And secondly, those contributing partners, then it's very much on them to basically submit the letter, decide what contributions they're bringing, and to ensure that they do in fact qualify as contributing partners and aren't in any way associated with the industry or private members of IHI.
Thank you very much, Desmond. Next question from Stefan. Not all animal testing is directly related to product research and development, but can also be related, for example, to testing linked to the manufacturing of already licensed products. Will non-animal testing related to testing of manufacturing also be in scope in this call?
So, Stefan, thank you for your question. This is a very good question. Yes, indeed, the production is also of health technologies is also in the scope of this topic text. And if you read carefully the topic text, you will understand that manufacturing of health technologies and animals or animal-based material used for this to this end is within the scope of the topic text.
Fantastic. Thank you so much, Salome. Next question. Is 30 million the total budget for topic?
Well, so the total available IHI budget for this topic is 30 million euros. But as we mentioned previously, we estimate that about 12 to 15 millions of IHI funding should be sufficient per project, but only if you have these amounts, 12 to 15 million euros of contribution from the project participants. So globally, the total budget per project, this is an estimation, could be around 24 to 30 million euros, but this is a collective funding, let's say, coming from the IHI financial contribution and the contribution from project participants. But as I already said, so we have 30 million of public funding available. We estimate 12 to 15 millions to give per project. However, the number of projects that will be selected, because this is the next question, how many projects will be selected,
Indeed,
will depend on the proposals received. So as far as they are above the threshold and they are highly scored, and the funding, the requested funding is justified, we will have accordingly a project number of proposal. Sorry, an eligible. Sorry, I will start again. So the number of projects that can get funded will depend on how much public funding you are asking, if you are above threshold and if you are highly scored. So there can be more than one project.
thank you very much. A question from Frank and addressed to Desmond. How is SME defined in terms of company size? Yeah, hold on a second, Desmond. You were muted.
I'm not going to take it personally that the lawyer keeps getting muted. That's pretty normal. Thanks. That's actually quite a good question. So SME, I mean, I think there's two issues here. So in its simplest way, SME basically has a defined size in terms of annual turnover, and how IHI basically approaches this is that IHI actually puts SME with also what we call mid-sized companies, and we basically say in the legal basis that generally companies with an annual turnover of 500 million or less can receive funding, but because this is a single stage call, it's actually possible for larger entities to actually receive funding as well. So the SME, I would refer you to the general EU definition just as a first point, but for this particular call, I think it's more important that you basically look, are we an identified independent legal entity? Do we come in terms of the other legal requirements? And if so, then you should, as one point of principle, be eligible for funding. When I talk about the other legal points or the other requirements, basically, are you coming from the EU or an associated country? If it's useful, we talked about this a lot yesterday in the Rules and Procedures webinar, and I know Salome referred to it earlier on, and I would refer everyone to that webinar. I think we took about 2 hours yesterday to go through all of these eligibility conditions and which kind of companies can actually apply for funding in this particular call. And you will find that extremely useful in addressing this particular question.
Thank you very much, Desmond. A question from Jeroen. Yesterday, though, it was mentioned that contributing partners cannot be in any way associated with the industry associations. If a company that wants to contribute is associated with one of the associations, can they still come on board?
Yeah, sure. So the idea of the contributing partner is that a contributing partner is defined as as a company or a foundation that wants to contribute in kind, but is not one of the private members. That is not one of the actual trade associations. And that's actually what it is. The reason why the contributing partner program has been developed is because we had something similar under IMI2, which was extremely successful, whereby these legal entities, for instance, some very, very big funders from the US and some other EU-based funders as well, foundations like the Simon Foundation, like the Gates Foundation, all contributed hundreds of millions of euros in kind. So the contributing partner cannot be associated to industry, as we just said, that's fine. Can't be associated to one of the trade associations, that's fine. But if you are indeed associated to one of the trade associations, that's fine. You can still join an applicant consortium. You can still contribute in kind. What will happen is that you will basically be noted as an affiliate of one of the trade associations. So you'll be classified as either what's called a constituent entity or an affiliated entity. Your in-kind will count towards that trade association's contribution of in-kind towards IHI-JU, and you'll be able to join the project. So that is definitely possible. So just because you are associated with one of the trade associations, you are not by definition excluded at all. In fact, you're very welcome.
Thank you, Desmond. I think Elisabetta wanted to add something.
Yeah, I just would like to give a little addition from, let's say, from the legal explanation of Desmond. So let's say you are an industry, whatever size industry, and you would like to participate contributing your in-kind to a proposal. Then the first thing you have to check is that as industry, you are not a member of the European Association, but very importantly, you should also check if you are not a member of the different national industry association, which are under the umbrella. So you should go to and sometimes you really don't know. We find out about that. So you should really go to the website of the type of industry trade association that better fit your type of industry. Check if there is any, or if there is also an industry national organization, you might be a member, because then in that case, you are perfectly eligible to provide your contribution without having let's say the extra burden to have also to have to apply to a contributing partner, because you are already, let's say, in the club. You are already a member of the IHI partnership. Instead, of course, if you are not that, then you will have to follow the way of the contributing partner. And please, as Desmond said, read all the documents and also look at the webinar on rural agriculture. So thank you.
Very good. Thank you so much. A question from Doris. Does a consortium need to have a contributing partner?
No, absolutely not. I'll refer everyone to what Salome said, which was basically the 45% in-kind contribution is completely non-negotiable. So that is absolutely essential that you hit the 45%. Obviously, as Salome also said, we recommend that you aim for 50% instead. There are various ways through which you can basically hit that minimum eligibility criteria. Contributing partners is certainly a popular approach, but it may not always be appropriate. So you have that flexibility there. It's something that can be used, but there is no rule which says you must have a contributing partner in each applicant consortium.
Thank you, Desmond. Julio is asking, when is the call open and closed?
Okay, thank you, Julio, for your question. So we have indicative timelines because, as you know, we are in a situation that the topic texts have not been approved yet by our governing board. So the indicative timelines is that end of July we will have the call of the launch of the call. And then in the mid-January, we will have the call submission deadline. In February, we'll have the scientific evaluation. End of March, we will inform applicants of the evaluation outcome. And finally, we will have this will end with the grant agreement preparation, grant agreement signature in June 2024. So these are, of course, indicative timelines and can be found in the slides of the rules and procedures that have been presented yesterday.
Fantastic, Salome. Thank you very much. Next question from Barney. So it might just be 2 projects that are funded in total, each at 15 million euros, for example?
Yeah, exactly. So of course, again, this is an estimation. But as we said, we estimate that they will need per project 12 to 15 million. So if it is 15 million, indeed, it could be 2 projects funded.
Thank you very much. Andras is asking, as this is a single stage call, the IHI FPA industry partners need to be contacted and fully integrated into the proposal plans, all of them?
So I'm not totally sure to understand your question, but indeed, this is a single stage call, and you need to create a consortium, including ideally the industrial partners, not only FPA. There are several of them because, yeah, the IHI is a cross-sectoral collaboration. But you can also reach out to companies of FPA or companies of the other industries associations. I hope this clarifies, Andres.
Thank you very much. So that's it for the moment. While we wait, I would like to promote a little bit the next info sessions that we're holding. The next one is actually at 2 p.m. today, Therasnotic Solutions. Then we have tomorrow stroke management. And on Thursday, we have 2 sessions, one on synthetic data generation and one very important about the financial part of the proposal at 2 p.m. And we will explain a little bit there how to present a successful financial proposal. Also, just like a reminder that this recording is going to be available in our website in ihi.europa.eu and as well as in the B2Match platform. So if you missed the part or if you got disconnected, you can watch it again as many times as you want by clicking on the link to our YouTube channel in our website. So I think we don't have any more questions. So we're going to go ahead and have a small break of 5 minutes, and then we will start with the pitching session. We have 9 pitchers in total, and they will dedicate 4 minutes to enthusiastically sell their project ideas. So please stay with us and don't go anywhere. Well, you can take a break for 5 minutes, and then we will resume. Welcome again. We're back from the 5-minute break. And now we are going to start with the pitching session. And now we're going to start sharing the presentation with the list. So we're going to be calling the pitchers in order of appearance. So whenever you're called, please unmute yourself and start with the pitching of your project idea. All right, so the first pitcher of today's session is Geir. So Geir, please, if you're there, unmute yourself and you can start with the presentation. Okay. All right, Geir.
Hello, everybody.
Hello, we can hear you. Please go ahead.
Yes, will you bring up the slides, please?
Yes.
Yes, thank you. Excellent. Hello, my name is Geir Klinkenberg, and I'm a research manager in a group called Medical Biotechnology in an organization called SINTEF. SINTEF is a large not-for-profit research organization with main offices in Trondheim in Norway. Next slide. Yeah, thank you. So our project is focusing on standardization of in vitro and in silico efficacy and safety models for the screening and prediction of drug effects. The take-home message is that ATMPs, novel drugs and formulated drugs, are poorly characterized by current in vitro models, as they frequently interact with the scaffold material used in such 3D models. There is a high need for validated and standardized animal material-free 3D models, and this will be delivered to be used for drug efficacy and safety testing. We aim to provide organ-specific tissue models that can represent physiological relevant uptake and exposure kinetics and yield meaningful readouts for drug development and evaluation in a high-throughput manner. Next slide, please. So our main activities is to standardize 3D multicellular models that recapitulate organ-specific characteristics and variables, to develop and standardize immune-enhanced in vitro assays, implement barrier models for the most common exposure routes, skin, lung, and gut, to develop in silico and digital twin models, and demonstrate transferability of the developed NAMs. Next slide. So what we offer is an interdisciplinary consortium with competence within organ-specific and immune models, high-throughput screening, standardization for regulatory preclinical documentation, material development, analytical characterization of drugs and materials, and state-of-the-art infrastructure for screening, material process and drug development and productions. We are no ICOP or IHI organizations in the consortium so far. Next slide. The expertise we are seeking is SMEs with interest in NAMs, readout technologies, or use cases. SMEs or companies working on new animal material-free products. Large companies interested in testing and implementing new NAMs, regulatory bodies and policymakers, and insight in market needs, requirements from new NAMs, and industry user knowledge. I think that was the last slide. So thank you for the opportunity.
Thank you very much, Geir. Thank you very much for your presentation. The next speaker is Margarida Diogo, assistant professor at the Institute for Bioengineering and Bioscience, pitching the hiPSC technology for engineering human tissue models. Margarida, I'm going to mute yourself. I'm going to mute you. Please start when you want. Thank you.
Good morning, everybody. So my name is Margarida Diogo, and I am a senior researcher at the Institute for Bioengineering and Biosciences in Lisbon, Portugal. And I'm here to talk a little bit about our expertise and competences regarding the use of the technology of human-induced pluripotent stem cells to engineer human tissue models, which I think it is very relevant in the context of this topic 1 of Call 5. Next slide, please. So as we all know, the use of animals in biomedical research and development raises serious ethical issues. We all know that animal testing requires time-consuming protocols, high costs for animal supply, and more importantly, the results are not always reproducible and applicable to humans. So in this scenario, we believe that it is crucial to develop alternatives, allowing significant reduction of animal usage and animal suffering level, and also allowing a more trustful recapitulation of human organs physiology. So we believe that we can contribute to this endeavor specifically by establishing non-animal approaches based specifically on the use of human organoids derived from human-induced pluripotent stem cells for the efficient development and testing of safe and effective innovative therapies. In terms of applications, we believe that these non-animal approaches may be used specifically for modeling infectious or non-communicable diseases, for drug testing, and for toxicology assays. Next slide, please. So in this pitch presentation, we are presenting ourselves as a possible partner, not a coordinator, but we actually believe we have an overall idea of how a wider project could be designed for this specific topic. So in the first place, we should select a disease or a condition and an affected organ, and then we should pursue with a very deep and in-depth characterization of the currently available models, including some human samples, using omics technologies with the head of computational approaches. Then we would start with the development of our humanized models, specifically our organoids, and then we would translate the culture of the organoids to a body on a chip or organ on a chip platform to allow a more trustful recapitulation of the human physiology. After developing these humanized models, we would once again perform a very in-depth characterization of these new models using, for example, single-cell RNA sequencing to validate our models and be sure that they are actually recapitulating the human physiology. Of course, that after this validation, we would also have to work on the standardization and the regulatory aspects. Next slide, please. So in this possible wider project, we think that our expertise and resources are very useful, namely because in the last few years we have been working in the reprogramming and gene editing of human-induced pluripotent stem cells, both from patients and from healthy individuals. We have also been working on engineering from scratch novel cardiac, cerebellum, and hepatic organoids, and we have been also working on modeling neurological disorders using brain organoids and assembloids, in particular in the context of neurodevelopmental disorders. Next slide, please. Moreover, we have been also working on establishing in the lab a portfolio of molecular, structural, and functional methodologies for in-depth characterization of our organoids, including bulk RNA sequencing, single-cell electrophysiology, microelectrode arrays, confocal microscopy analysis, and calcium transient analysis, among many others. Next slide, please. Finally, of course, our expertise would not be enough for such a wide project, so we believe that we should bring together to us experts in machine learning, data analysis and integration, organoids on a chip, and also standardization and regulatory aspects experts. And this would be my last slide. Thank you very much for your attention.
Thank you very much, Margarida. So the next one is Gael Roue. He is a group leader and EU-Interreg project coordinator at the Josep Carreras Leukemia Research Institute, presenting implementation of a new immunocompetent non-animal PDX model of hematological cancer for immunotherapy drug discovery. Gael, please, the floor is yours.
So good morning, everyone. I'm Gael Roue. I'm the head of the Lymphoma Translational Lab at the Josep Carreras Leukemia Research Institute in Barcelona. Today, I will present a project titled Implementation of a New Immunocompetent Non-Animal PDX Model of Hematological Cancer for Development of Immunotherapeutic Drug. So, next slide, please. So the current limitation of the available mice PDX model used in the hematological cancer research are first, the reduced engraftment rates, since we obtain in the case of aggressive B-cell lymphoma less than 40% of success using immunocompromised mice. This process is a long process and it's very costly and requires a large number of mice with several generations to obtain a stable PDX. These models are based on fresh material, so you need a fresh, in the case of lymphoma, for example, fresh lymph node, which is quite challenging with the current diagnosis procedures. And these models are not, excluding in the case of humanized PDX, are not suitable for development of immunotherapeutic agents. So in the project, we propose new solutions based derived from the chicken embryonic coordinatory membrane, the CAM assay. So this model, which is a non-animal model, take advantage of the rich vascularization of the chicken embryo development. This is a low-cost and highly efficient model, which is quite more efficient than the mouse PDX model. You need a very low input of primary materials. You can work either with fresh or frozen materials. It's a natural immunocompetent, since after a few days of embryonic development, the chicken immune effectors are functional in this model. The engraftment rate so far in the case of acute leukemia and aggressive B-cell lymphoma is close to 100%. And it's a very quick process, since in less than 2 weeks, you can have all the design information and toxicity, genomic, proteomic profile, and efficacy of new drugs. And in the lab, we have developed more than 15 readouts to evaluate these features. The reproducibility is increased compared to mice model, and of course, the non-animal in vitro model that allows you to comply with the 3 rules. Next slide, please. So what we propose is to bring access to PDX models from hematological patients derived from these CAM models, basically for development of immunotherapeutic drugs. Since it's an immunocompetent model, this technology is already available in your lab and have been validated in the last year by 5 publications in top journals in different models of acute leukemia and aggressive B-cell lymphoma. Next slide. So here you have summarized basically the workflow of the lab. In this case, we have you can see how we processed with the primary lymph node biopsy. So from this biopsy, we can develop either the CAM model, which is on the right side, and we can run the side-by-side comparison with the classical PDX model in mice, but also a multicellular spheroid model, or in 2 years from now, we'll have access to the lymph node on a chip model to compare different models. So with this different in vitro and vivo procedure, we can assess the different profiling of the tumors, response to drug, or small molecule inhibitors, antibody-based therapy, or cell-based therapy by a single-cell multiomic analysis, and including epigenetic analysis, thanks to the availability of the primary samples available in the biobank. Next slide. And what we would need from the consortium is some industrial or academic partners interested in the access to this in vivo avatar, which are physiologically reliable. with access to the artificial immune system, which allows you to evaluate the efficacy and the safety of new treatment, antibody, small molecule inhibitors, and cell-based therapy, depending on the tumor microenvironment. Thank you.
Thank you very much, Gael. Our next speaker will be Aydanur Aydin from the Gumushane University. Unfortunately, he will not be able to deliver his speech, so we will skip this presentation and we will go directly to the following speaker, who is Frank Bringezu. So Frank, he's a principal scientist at Merck and will be delivering his presentation, ViCoGs to replace concurrent controls in animal studies. Frank, please, the floor is yours.
Yeah, hi, hello. This is Frank Bringezu. I'm working as principal scientist at Merck Healthcare in Darmstadt, and I'm here on behalf of the ViCoG team that actually is proposing here a project on virtual control groups to replace concurrent controls in animal studies. Next slide, please. What are the challenges and objectives that we actually have identified? So the project aims to build virtual control groups to replace concurrent controls in animal studies. This is because of ethical reasons to reduce animals, but there are also other reasons mentioned on this slide. First of all, as you know, that in toxicity studies, there's always a control group present, and this may open the opportunity to reduce these used animals and these control groups by 25%, which is a significant contribution to 3Rs. There's also a cost-saving aspect because if you imagine that a non-human primate costs 40,000 euros per animal, then there is a high impact if you can reduce the controls in the toxicity studies. The project is fully in line with the objectives to reduce animals, and it is across different industries a valid proposal, which means we can actually have different context of uses because toxicity studies are performed for pharma, but also for other purposes. And here we see a great potential for integration of these methodologies. And finally, we will also be able to support AI-driven applications, for instance, for the creation of virtual animals or so-called digital twins. Next slide, please. Our main activities are on this slide summarized. We will develop a validated database for the virtual controls. We will provide statistical procedures for retrospective and prospective evaluation of the performance of virtual controls. We will develop matching procedures for the qualification of virtual controls, and this all will be done in close collaboration with regulatory authorities from the very beginning on to achieve acceptance from the regulatory perspective. The proposal, how this approach will be implemented and developed, is published in Altex. You can see the publication here, and the workflow is depicted in this small graphical schema, which illustrates how we extract the controls from concurrent or legacy studies and develop a cloud-based application that eventually opens the way to statistical analysis across different studies, from different vendors, and finally will open the door really to establish a virtual animal at the very end, or a virtual control. Next slide, please. What we have on expertise and resources is summarized here. We have formed a pre-consortium already with FPR and associated partners that may contribute the ICOPs and the IKAAs. And here you see the different pharma partners that actually have signed agreements to contribute to the project. And this compiles a long list of, let's say, pharma companies, but includes already also chemical companies, like for instance BASF, going in the direction of integration of these approaches across industries. We have also small medium enterprises and academic partners identified already, and this is shown in the middle column of this small table. And we have also outreach to regulatory authorities, here in particular to EMA and FDA. And FDA is contributing to FUSE, and FUSE is already has already indicated high interest to contribute to the project. The in-kind contributions that we have already in the project, in this pre-consortium, include control data from different tox studies, from in vivo studies, and the current estimates amount to more than 1,000 GLP studies with more than 30,000 animals. And we are talking about roughly an in-kind contribution of about 10 million euros. We have compiled a pilot database that contains these data in a so-called SEND format, which is a submission relevant format. And this is a good starting point to start with all the activities. Next slide. Having said what our expertise is, now this is on the expertise that is requested. And you may imagine that if we want to use controls in a validated, let's say, GLP system, then computer system validation is one very important topic that we are looking for, and we are looking for expertise in this regard. Statistical support is very important, cloud-based database development as well, data visualization, digital pathology, because everything that we do includes not only in vivo data, but also pathology evaluation, and digital pathology is a very important asset here. And for sure, we are looking for sustainability of this approach, and this is something that we are looking for. And possibility for regulators is here completely.
Your time is almost over. If you could speed up a little bit, thank you.
It's now the next slide, which is probably the last. So the outlook, the project requires close collaboration, as I mentioned, and we are looking across different regulatory authorities. It's the systemic toxicity that we first focused, but we plan to extend, and we have already outreach to RITA and to NTP in this regard, and we see a strong collaboration between different IHI projects here. Big pictures mentioned. With that, I'm at the end. Thank you so much.
Thank you very much, Frank, for your presentation. And now the next speaker will be Eva Gonzalez. Eva is the head of Innovation Department at BIOLAN and will be presenting in vitro selection of nanobodies for healthcare applications. Eva, the floor is yours.
Good morning, everybody. My name is Eva Gonzalez, and I'm going to briefly introduce you to the project in vitro selection of nanobodies for healthcare applications. Next slide. So BIOLAN is a Spanish SME focused on the development of analytical tools for the agri-food and healthcare sectors. And the objective within this project is to select and produce synthetic nanobodies as recognition elements for developing advanced diagnostic devices for healthcare applications. So we want to overcome the use of animals for the production of molecular recognition elements such as antibodies and nanobodies. And we want to do this by delivering synthetic molecular recognition elements with enhanced characteristics. So as fast screening, high batch-to-batch reproducibility, high specificity and affinity, and high stability. Next slide, please. So the main activities, project activities that we envision is that once we identify the target or targets analyzed, we want to select the nanobodies against these specific targets. We will characterize these nanobodies and then we will apply them as molecular recognition elements for healthcare applications. Next slide, please. The expertise and resources that we offer. So we have developed a platform for delivering nanobodies able to recognize any potential antigen. We do have a synthetic nanobody library. We have defined screening protocols with no ViCoGs required. We perform the expression on E. coli and we also run purification. The platform has been already validated with the selection of a nanobody specific for lysozyme. And we have 2 nanobodies under development at the moment in the pipeline. Next slide, please. And we are looking for a consortium interested in the application of these novel synthetic molecular recognition elements for healthcare diagnostics. And the profiles we are looking for cover all the range from SMEs, large companies, and research institutes. And I think that's everything from me.
Fantastic. Thank you so much, Eva. The next speaker is Jerome Martinez. Jerome Martinez is a senior scientist at Bioengineering, and he will be presenting fetal bovine serum, compassionate alternatives for research, ethics, and sustainability. Jerome, please, the floor is yours.
Hi, everybody. My name is Jerome Martinez, and I'm representing today the Immunoassays Research and Development Department of Bioengineering, a French in-vitro diagnostic manufacturer. So, more specifically, I'm in charge of pre-formulation studies for immunoassays reagents and physicochemical investigation for the wide range of biomed solutions. And today I'm very excited to introduce you a very impactful ethical concern for Bioengineering and very probably for the whole biopharmaceutical industry. Next slide, please. As you probably know, as a major actor in in-vitro diagnostic market, our products need to meet the best possible technical performances in terms of sensitivity, specificity, and stability. To achieve such specification, many animal sources, raw material, and biomolecules have been used since decades in all reagents. Among all of these animal origin components, the fetal bovine serum, as known as the FBS, is a widely used additive for many kinds of biopharmaceutical products, such as vaccines, cell culture media, therapeutics, and in vitro diagnostics reagents. Anyway, behind the FBS, next slide, please, is hidden the several classic issues from the animal sourced components we regularly face, like the high batch-to-batch variability, the bad sustainability, in addition to possible fraud and contamination risk linked to this type of raw material. Anyway, all of this is almost minor consideration when you discover that the FBS is taken when pregnant cows are slaughtered by removing the fetus from its mother's womb, then all of its blood is taken by a cardiac puncture. This process is more often performed without any anesthesia in order to guarantee the quality of the sample. We believe that such practices are from another ages and specifically make the FBS a product of major ethical concern. Next slide, please. The market forecast for the FBS shows that its use is still growing worldwide and will reach in 2030 $1,500 billion, almost twice the market value of 2021. And as you can see, the European market has the same growth rates than the US market. Next slide, please. On the cell culture media market, many solutions are proposed today as alternatives for the FBS like the human platelet lysate or the heat inactivated coulombic fluid. These serum-free solutions for cell culture market are out of our project scope because the FBS mechanism involved in vitro diagnostics reagents are in many ways totally different. Even if since few years, all of the new immunoassays we perform in bioMérieux development are totally out or totally avoid the use of FBS, we still have today on the market many reagents which have been designed decades ago. And linked to our product lifecycle management, the replacement of the FBS needs to be at the same performance. So it's a big challenge for us today. So next slide. The main idea of this project proposition is 1, to clarify the components role of the FBS in the different ways to use it as a specific or as a non-specific blocker, as an enhancer for the IVD reagent, or any kind of other use depending on the IVD use. And 2, of course, trying to identify non-animal or even better, a fully synthetic substituent for each use case. Next slide, please. We strongly believe that this critical concern for our IVD reagent may be shared with many of you, and that's why we're looking at a strong partnership to overcome this ethical concern with any IVD player or biopharmaceutical companies, research institutes, startups, SMEs, and regulatory bodies. And we know that we may share some vision of a world where public health does not involve animal suffering. Thank you a lot for listening.
Thank you very much. Now our next speaker will be Simone Calzolari. He is the CEO of CEClinics, and he will be presenting Accelerating Human Safety Risk Assessment with Integrated Approach. Simone, please, the floor is yours.
Hello, thank you everybody. I hope you hear me well. So I'm from CEClinics, a zebrafish company based in Barcelona. So thank for me here. Next slide, please. So the purpose of the project idea is to tackle one problem, and the problem is the fact that 1/3 of drug candidates are toxic, and tox is one of the major contributors of costs in drug development, and even more if detected later in clinical and aftermarket phases. And second, of course, animal models are several publications that confirm that are not good predictors of tox, so alternative methods are needed in order to uncover problem of toxicity early in drug development. And of course, since we are in this topic, the ethical issues about the use of animals are more and more present in our society. Give a concrete example of our potential results. It's what we aim is to better predict the toxicity of new compounds in drug development, and of course, the complete eradication of the animal use. Next slide, please. And our proposal is centered in the model that we use for our research, and that is the zebrafish embryos. And the property of this model are several. It's transparent, all the development is ex utero, and so please in the slide have all different organs in the zebrafish embryos. So it doesn't appear, but I can explain. Have a liver, heart, central nervous systems, and you can see directly and visualize the tissues and the cells. And you, of course, can test several compounds at the same time as an high-fructose screenings. Somebody can say that this is an animal, but it's not, because it's published an EU directive that lists its embryos up to 5 days. That actually is in the time window where we normally make our assays are considered as an in vitro model and non-animals. So it's perfectly fitting with this call. And of course, when we use zebrafish, we contribute also in the second R, that is the reduction of animal use, because we'll use less animals compared to what are used in other applications like toxicity tested in mouse. and of course, in some application is not invasive, so contributes, I would say, and everybody says, to the 3 R's and is fitting perfectly in the call. Next slide, please. We have already made some steps in testing toxicity with zebrafish, and we have recently, well, 3 years ago, published a paper where actually we tested several compounds and the different outputs in terms of phenotypics in zebrafish, and we got improved results in terms of sensitivity, specificity, and accuracy compared the same results in mouse with a comparison with humans. So we proved, and I invite you all to check in that publication, the zebrafish is a good predictor of developmental toxicity, and that you can list here all the different outputs that are the comparison in humans. So zebrafish, with these slides, I want to confirm that it's a very good model for testing the tox and in general toxicity. Next slide, please. And so our company is not using only zebrafish. We have recently opened a new area that is dedicated to organoids, that is called the Neuroid. And the aim of the platform, the technology that is central, is to use human iPS cells in order to derive neurospheres, and with the neurospheres create networks. That is already approved technology, and you can see in the video. So click in the video, please. And the neurospheres only in the platforms and in plates are created networks, and then networks are indication of interactivity. So we can test compounds that actually affects the interconnectivity between neurons and between neurospheres. So that is one other platform that we have developed, close and complementary to zebrafish platforms, in order to test toxicity of compounds that's already proven, but also new chemical entities. Next slide, please. So the idea is to use zebrafish plus organoids and plus more tools that can be integrated on that in order to create an integrated approach for testing assessment. So we think the project like that. So initially we need non-testing methods like the listed here, all the different technology in order to discard, prevent, or in silico limit the compounds that will pass to the second phase, where actually the experimental systems that is combining the zebrafish testing and the organoids testing, and more tests that can be added to these platforms. And in high throughput levels, we look into the whole organisms, like in zebrafish case, also combining with the human biology, checking for the organoids technology, and of course contribute to the non-animal testings. Once we
Simone,
yes.
Time is almost up, please.
Yes. So the third phase is to check for the automatic screen technology and analyze combining phenotypics, transcriptomics in and extracting with the learning technology. And we hope to increase predictivity in order to limit the human risk. Next slide, please. So what we contribute on that is more than 10 years of experience in toxicology using zebrafish, high throughput screenings, the combination of transcriptometry, phenotypic, and AI tools, and exclusive top-notch human Bioengineering platform to study neural connectivity disease. Last slide. What we are requested, the expertise that we are requested are not testing methods like computational modeling, testing methods, but in human and zebrafish cell-based assay for neural tissues, omics, and ADME, and of course a contribution of regulators like experts in IHI guidelines and NAMS-regulated acceptance. Thank you very much.
Thank you very much, Simone. Now, last but not least, we have Daniela-Elena Costea, professor in tumor pathology at the University of Bergen, presenting tailoring ex vivo 3D multicellular models for use as predictive biological tools in the treatment of metastatic recurrent head and neck squamous cell carcinoma. Please, Daniela, the floor is yours.
Thank you. Good morning. Can you hear me?
Yes, we can. Please go ahead.
Thank you. So I am professor in tumor pathology, and I work both at the University of Bergen and at the Haukeland University Hospital. And my expertise and interests are in tumor host interactions and establishing 3D models, human-based models for research and better treatment of cancer patients. If we can go to the next slide, please. So I am very focused on head and neck squamous cell carcinoma. And what we present here is an idea that we have because we have developed ex vivo 3D multicellular models. That means that we have models in which we have cancer cells, we have cancer-associated fibroblasts and immune cells derived from the same patient. We have established these models, and now we are using them, and we try to see if we can use them as predictive biological tools that can be implemented into the current clinical scheme of treatment for head and neck squamous cell carcinoma. We are focused on this type of cancer because there are very limited treatment strategies, particularly for the metastatic head and neck cancer patients. And now in Norway and Western Europe and Americas, we see there is a pandemic of HPV-related head and neck cancers, but these models can be used for any type of cancer. So we think that these models can be used for the concept of personalized cancer therapy called clinical trial in a dish, where you could try different drugs on these mini tumors developed ex vivo, and then you could have this result at the tumor board, and this could help in taking a very informed decision for the treatment of that particular patient. So we have the model, and what we are thinking now is that we need to optimize them to have a pipeline to produce these models that can be used for multiple drug testing, and this could be done within 20, maximum 30 days from the time of biopsy, which is the time that, at least in Norway, it is mandatory to have a treatment within this time for a patient diagnosed with this disease. If we can go to the next slide, please. So as main activities in the project, we are thinking that we need to assess if this 3D, we think that these 3D multicellular models reflect and mirror the in vivo response to a certain drug in the patient, but we are not sure that is exactly how they will react. So this is what we first would like to test them to standard care drugs. As I said, there are not so many. At least in Norway, we have 5 different schemes of treatment, and that's why we need to include immune cells in our models, because also we want to include the immune checkpoint inhibitors. But we can use these models also for identification of new drugs that are currently available for other diseases or other type of cancers, but not particularly for this type of cancer. And as I said, we would like to optimize these models so it can be integrated in the so-called package that we have or we have to provide for these patients. Next slide, please. So we have expertise, of course, in the pathology. We have clinical oncologists in our team, and my lab is specialized in primary cell isolation. We recently got also ethical approval to isolate cells from the autopsy and from the bodies that are donated for education and research, and we managed to isolate cells up to 6 days post-mortem. We have long experience with in vitro 3D modeling, and we have established also imaging mass cytometry, the Hyperion technology. Next slide, please. So we are using well, we would like to be a partner. We are not looking at coordinating. We are too focused for this, but if we could be part of a bigger consortium, we would be interested in also analyzing our models by proteomics and other omics approaches. We are interested in collaborating with drug testing platforms and with other centers for a multi-center study. I think that was it I had for today. Thank you very much.
Thank you very much, Daniela, and thank you, every one of you, to have listened to all these very interesting topic ideas and these speeches. I would like to remind you that you can find these and many other project ideas in the marketplace, and I think perhaps it would be useful if I could perhaps share with you how to look for it. So basically, you just go to your marketplace, click in here, and you just select the project cooperation that you're seeking. In this case, you could actually just go to Call 5, Accelerating the Implementation of New Approach Methodologies. And here you see that you have 31 opportunities for you. So just search for the one that you're looking for, and yes, you can get a successful consortium. So remember that we are here for you, and you can always ask us questions, and you can use this platform that we have set up for you. So thank you very much. That's all that we had to say today. Remember that we have another session today at 2 p.m. About theranostics solutions. So please join us and discover a little bit more about this topic and the pitching session that will occur afterwards. So thank you very much, and have a lovely lunch break.